Eicosanoids are a group of hormones belonging to the local hormones, which are sometimes called tissue hormones. Arachidonic acid gives rise to three major classes of eicosanoids: prostaglandin, thermoxane, and leukotrienes. However, although eicosanoids constitute a large number of similar hormones, they can exert different effects depending on the organ they are produced in. Eicosanoid synthesis begins with fatty acids from the cell membrane. The most important of these fatty acids is arachidonic acid. On the one hand, prostaglandin H2 is synthesized from arachidonic acid, which is the starting point for the synthesis of other prostaglandins and thromboxanes. On the other hand, arachidonic acid also gives rise to leukotriene A4, from which additional leukotrienes are formed. Arachidonic acid is released on demand from the cell membrane. The most important enzyme involved in this process is phospholipase A2, in short PLA2. Generally, the initial rate-limiting step in eicosanoid synthesis is the liberation of arachidonate from phospholipids by phospholipase A2, PLA2. The released arachidonic acid is converted to prostaglandin H2. The key enzyme involved in this conversion is cyclooxygenase. There are two isoforms of cyclooxygenase, cyclooxygenase 1 and cyclooxygenase 2, also termed COX-1 and COX-2. Isoenzymes catalyzes the same reaction but differ slightly in their structure. COX-1 is present in almost all cell types and is constantly active, whereas COX-2 is only expressed under certain conditions, such as an inflammatory response. Lipooxygenase catalyzes the reaction of arachidonic acid to leukotriene A4. As eicosanoids are local hormones, it is important to consider their physiological effect depending on the organ they are formed in. Prostaglandin E2 is a mediator of pain and inflammatory response. This effect is due to binding of prostaglandin E2 to free nerve endings called nociceptors, which register pain signals. It is also produced in the hypothalamus in response to chemical messengers released during inflammatory response. Therefore, prostaglandins increase the body's core temperature. In the stomach, prostaglandin E2 has a protective effect by facilitating mucus production. In uterus, it stimulates uterine smooth muscle contraction with increasing gestational age. Thromboxane A2 is released by activating platelets, resulting in vasoconstriction and platelet aggregation. Leukotriene B4 is synthesized by immune cells, especially neutrophils, to attract other immune cells, indicating that it has a chemotactic effect. Excess production of leukotrienes can result in an asthma attack. The bronchoconstrictive effect of leukotrienes can be reduced by blocking the receptors of leukotrienes. Do not forget to like the video and subscribe to our YouTube channel.